Hey guys, so the last thing we have to cover for this year, we've reached the end, is something known as biotechnology. So far we've talked about natural ways for you to get specific traits, right, through sexual reproduction, through mutations. Those things cause differences within a population, but those all happen naturally, right? We have nothing to do with that. The last part of this we call artificial selection, artificial meaning like fake. So, using biotechnology, right, technology and science with biological organisms, we are manipulating or changing the genes and we're choosing the genes that we want to see produced. Okay, so in this, humans are selecting the genes that they want to see. Okay, so if you're looking for a partner, like some things that you would probably look for: maturity, honesty, respect, and independence, sense of humor, right? So you are choosing the traits that you want in a person, right? Okay, so we've seen this um, throughout history. This is an Old English Bulldog. This was from, I want to say, 1800s. So it's known for its strong jaw, which stuck out below the other, its speed and intelligence. Then you also have the English Mastiff, which is known for its size. It is the largest breed of dog, its loyalty and distinctive black face. So, through a process called selective breeding, okay, you are able to breed these two organisms together to get one organism that has both of the traits, right? So then if you did that, you would get what's called the bull mastiff. So it's known for its strength, size, loyalty, intelligence, and instincts. So it has the traits from both of these in one organism. Okay, so again, that's called selective breeding. Okay, we breed animals together to get the traits that we want. Unfortunately, there often comes some disadvantages with this. So this type of dog, if you notice, he has these folds in his skin where bacteria can start to develop, and he has a skin condition called atopic dermatitis. So if you have this type of dog, then you would have to really clean in between the folds so that that bacteria doesn't build up over time. Also with the German Shepherd. Sorry, I blanked on the name of this dog. German Shepherd. Years ago, they used to have a flat back like this, but it's a police dog, often used as a police dog. So they've been taught to sit back on their hind legs, and as a result, they have a lot of hip issues and problems with their hips and hind legs. Okay, and this I just think is so adorable, and I would totally want one, but they're super expensive. So this is called a Pomsky, and you would take the sperm or egg out of a Pomeranian, this little guy here, and you would breed it with a Siberian Husky. And when you breed them together, you get this adorable little Pomsky, which is the size of a Pomeranian, but looks and has traits of a Siberian Husky as well. Okay, so you wouldn't have to necessarily physically mate them together. You could just take the sperm out of one, the egg out of another, and then put it into a female, and then it would produce this Pomsky. Again, super adorable. We've seen this with horses too. So if you follow horse racing, they'd want a fast horse, a muscular horse, so that there's chances of them winning, right? So they would find horses that have those qualities and breed them together so that you would have one horse with multiple of those qualities. Like chickens, back in 1957, chickens looked like this and weighed a lot less. And they probably realized over time that if a mutation happened or through just sexual reproduction, one chicken would get bigger. So over time, they would breed the big chickens together to get a really, really big chicken. So that would be beneficial because then they could sell more meat from the chicken and obviously make more money that way. So they've bred the chickens to be larger over time. Okay. So again, that was called selective breeding. The second one that we're going to talk about is called genetic engineering. This is when you take a gene that you want out of a human. Okay, so you cut a specific gene out of human DNA and you actually paste it, it's almost like a cut and paste, into a bacterial cell. And then you put it back into the bacterial cell, the bacterial DNA into the bacterial cell, and then the bacterial cell is able to make multiple copies, and it's all going to have this gene inside of it. One reason why we would do this with humans is so that we can make insulin. So if you remember from the diabetes lesson, then you remember that diabetics either can't make insulin or are resistant to insulin. So they would need to take a shot of insulin or get it from someplace else. So we would take the gene out of a human, the insulin gene, and we would put it into bacterial DNA, and then we would make copies of that gene, and we would have multiple copies of insulin. The reason we use bacteria is because, if you remember, it reproduces very, very quickly, and it reproduces asexually, so they're all going to look identically the same, and they'll all have this gene. They've actually genetically engineered glow-in-the-dark pigs. So there's a fluorescent jellyfish that glows in the dark, and they took that protein out of the jellyfish and put it into a pig embryo. So when the pig got older and it goes under black light, it's now going to glow in the dark the same as the jellyfish would. Okay, so that's called genetic engineering. The last part of biotechnology that we'll talk about is cloning. So you probably know that a clone is something that's identical, but the process is actually really complicated, and I'll do my best to explain it here. So you have a donor cell taken from a sheep. Okay, this is the reason they say sheep is because Dolly was a sheep and she was the first successfully cloned animal. Okay, so you take a body cell out of one sheep, okay, and you remove the nucleus. Okay, then you put that into an egg cell of another sheep. So basically now you have a zygote. You have an egg cell that's pretty much been fertilized with another sheep's nucleus. And then you put that embryo, the fused cell that develops into an embryo, back into the foster mother, the other sheep, and it will develop into a genetically identical cloned lamb from this sheep. It's not this sheep because this nucleus was removed. So all the DNA is from the donor nucleus that we then put to the egg. I know, very complicated. But that's cloning in a nutshell. So selective breeding, when you take two organisms, breed them together to get the traits that you want. Genetic engineering, you take the specific gene out of an organism to get the gene and trait that you want and put it back into bacteria to be copied. And then cloning, you take a donor nucleus out of one organism that you want to clone, put it into a surrogate or foster egg cell of another female, and then it grows a genetically identical clone. Okay, but with all three of these, there's advantages and disadvantages, which is what your assignment is on today. Have fun!